Hi, I'm David, and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. 1A shows some steps in glycolysis. 1. Explain why glucose is phosphorylated at the beginning of glycolysis. Phosphate groups are transferred from ATPs to glucose to form fructose 1,6-bisphosphate. The phosphorylation activates glucose. The energy level of glucose increases after the addition of the phosphate groups. It becomes reactive and unstable. This is essential for the next reaction when it lyses to form two trisphosphate molecules. Besides, the addition of phosphate changes the shape of glucose. They cannot leave the cell using the transport protein as they are no longer complementary. Lastly, its concentration gradient is maintained when glucose is converted into something else inside the cytoplasm. This causes glucose to enter the cell continuously. 2. Suggest one use of reduced NAD that is produced in glycolysis. NAD acts as a hydrogen carrier. It can provide hydrogen ions and electrons to the ETC for oxidative phosphorylation and chemiosmosis to take place. It can be used in ETC and other redox reactions as it can donate electron to reduce another molecule when it is oxidized. If anaerobic respiration takes place, reduced NAD releases its hydrogen to reduce ethanol or pyruvate. ATP is made in glycolysis by substrate link or substrate level phosphorylation. It is the production of ATP from ADP by a direct transfer of a phosphate group from a phosphorylated compound. Hydrogen carriers such as NAD, ETC, and chemiosmosis are not involved. B. Describe the main conditions that are required for pyruvate to enter the mitochondrion by active transport. A molecule uses active transport to pass through a membrane when the membrane is impermeable to it. That is why carrier proteins are needed. These carrier proteins are found on the membrane of mitochondria. Active transport is used to move a substance against its concentration gradient. There is a lower concentration of pyruvate outside the mitochondria, which leads to the need for active transport. Otherwise, they can enter by facilitated diffusion. Pyruvate does not enter mitochondria if aerobic respiration does not take place. This is because the link reaction and Krebs cycle would not be operating. So, oxygen is available when it moves into the organelle. Lastly, energy is required for this movement. So, ATP must be available. C. Describe the link reaction. First, decarboxylation occurs. A carbon atom is removed from pyruvate as carbon dioxide. Then, pyruvate is oxidized. Dehydrogenation takes place. The removed hydrogen is accepted by NAD. A reduced NAD is produced. The remaining two carbon fragments, called acetate, will combine with a coenzyme A. Acetyl-CoA is formed. 2A wants you to explain the meaning of haploid and diploid. Haploid is one complete set of chromosomes. It contains one copy of all the genes needed by an organism. Do not say that haploid is half a set of chromosomes. Diploid is two complete sets of chromosomes. A diploid cell contains two copies of all the genes required by the organism. You should not use the haploid and diploid numbers for human to answer this question. This is a general question about the terms, not specifically for any species. You also shouldn't refer to gametes and somatic cells in your answer. Even though many species have haploid gametes and diploid somatic cells, there are exceptions such as bees and ants. So, these cannot be used as a definition for the terms. B. Describe the behavior of chromosomes during the main stages of meiosis I in animal cells. The question is only about the chromosomes, so do not discuss about the other structures such as the nuclear envelope and centrosomes. During prophase I, the chromatin condenses. They become shortened and thickened, and this is why chromosomes become visible. Then, the homologous chromosomes pair up. Each pair is called bivalent. Crossing over occurs between non-sister chromatids. The point of contact between the non-sister chromatids are called the chiasmata. In this process, the exchange of genetic material between the non-sister chromatids occurs. In metaphase I, the homologous chromosomes or the bivalents line up on the equator of the spindle. You can also call the equator metaphase plate. Independent assortment of homologous chromosomes occur. It is when the homologous chromosomes position themselves either way up on the equator of the spindle fiber. so they segregate randomly. The homologous chromosomes will then separate and move to the opposite poles of the cell in anaphase 1. In telophase 1, the chromosomes reach the opposite pole. This is where you can find the centrosomes. The chromosomes decondense and chromatin forms. They will not be visible again after this. The division of cytoplasm is called cytokinesis. The cytoplasm of the parental cell divides into two daughter cells. Note that this is a stage in the cell cycle, but it is not considered as a part of the nuclear division. Question 3a is about the fruit of a plant called seed blush. The seed can be winged or wingless, determined by a single gene with two alleles. The type of variation shown by the fruit wing is the discontinuous variation. This is because there are two categories for this phenotype. There are no intermediates between the two groups. 2 wants you to suggest 2 ways in which a new fruit color could naturally occur in a seed blush population. Scientists generally accept that mutation is the reason that leads to the formation of a new allele. When there is a spontaneous change in the nucleotide sequence of a gene, the polypeptide coded by the gene may be altered. It can result in a protein that gives a new fruit color to the species. Genotype is not the only reason that affects the phenotype. A new trait may arise due to a change in the environment. A new selection pressure causes the organism to change to adapt better to its environment. Cross-pollination may have occurred. This population may crossbreed with another population with a different fruit color. The hybrid they produce will show the new fruit color not originally found in the population. It is also possible that the new allele introduced to the population is co-dominant with the existing allele. So, a new phenotype is shown by the offspring that are heterozygous. A3 suggests 3 examples of the evidence obtained that help to confirm that these seed blush plants belong to the same species. To answer this question, you have to use the definitions of species. The first and most common way to define a group of organisms as the same species is that they can interbreed to produce fertile offspring. This means that they are not reproductively isolated from each other. The second way is to use the concept of niche. Niche is a species' ecological role determined by how it interacts with abiotic and biotic resources in its habitat to survive and reproduce. If 2 organisms belong to the same species, they should occupy the same niche. The third way is by using the similarities between the organisms. The same species should have similar morphology, physiology, behavior, and biochemistry. You can also get marks by elaborating on the biochemical similarities. Biochemistry refers to DNA sequences and proteins. They should have similar DNA sequences or genomes. You can also say that they have the same genes. Since they have similar DNA sequences, the amino acid sequences of their proteins are similar too. B. Explain why natural selection occurs in populations. There are a few prerequisites for natural selection to occur: overproduction, variation, and selection pressure. All populations have reproductive potential. They can produce many offspring. Because of this, competition between the members may occur. They compete for mates, food, and other resources. These environmental factors act as the selection pressure. Selection pressure is an evolutionary force that causes a particular phenotype to be more favorable in certain environmental conditions. The selection pressure acts differently on the members due to genetic variation in the population. Variation can be caused by mutations, sexual reproduction, gamete formation, and random fertilization. You can get this marked by mentioning any one of the sources of mutation. Due to variation, some individuals have advantageous adaptations. They have the phenotypes or traits that provide them a greater chance of survival and reproduction. These individuals are said to have selective advantage. They pass on their advantageous allele to their offspring. The advantageous allele frequency increases in the population, leading to a change in the gene pool. The population will now be more adapted to the environment, and this is evolution. 4a. Name 2 domains that are a source of restriction endonucleases. They are bacteria and archaea. They use restriction enzymes to defend against bacterial viruses called bacteriophages. The enzyme prevents replication of the phage DNA by cutting it into many pieces. Many students made a mistake by naming the domain prokaryotes. Archaea and bacteria are the two domains of prokaryotes. Prokaryote is not a domain. B talks about a method to produce restriction endonuclease using E. coli. According to the description, recombinant DNA technology is used for the production. The question wants you to suggest and explain the steps needed. First, we need to obtain the gene coding for the specific restriction enzyme we want to produce. There are several ways to get the gene. You can extract the DNA from E. coli. You can also obtain the mRNA from the bacteria and use reverse transcriptase to synthesize the cDNA. Another way is to make a synthetic gene using nucleotides. Then, PCR will be used to make more copies of the gene. PCR stands for polymerase chain reaction. It is a technique for rapidly amplifying a specific DNA. Next, we need to get a vector. Since the host cells are bacteria, we will use plasmid as our vector. Cut the plasmid with a restriction enzyme to give sticky ends. After adding the gene into the plasmid, combine them using ligase. DNA ligase seals the nicks between the gene and the plasmid by forming phosphodiester bonds. When ligation is done, we now have the recombinant DNA. The recombinant DNA can now be introduced into the host cell. This is called transformation. Not all host cells would be successfully transformed, so we will use marker genes to identify those with recombinant plasmids. The transformed bacteria will be transferred to a fermenter and cultured on a large scale. When they express the gene, restriction endonucleases will be produced. We can now extract and purify the enzyme. There is a mark for AVP. For example, you can talk about a method of transformation such as heat shock treatment in the presence of calcium ions or electroporation. C. Describe the advantages of database for the study and use of restriction endonucleases. This question specifically asks about the study and use of restriction enzymes. So, do not answer using the general roles of databases in biological research. A large amount of information, such as the amino acid sequences of restriction enzymes and the DNA sequences of their genes, can be stored in the database. This information can be shared and accessed quickly by people who need it, since it can be stored online. Comparison of sequence data can be done easily. Software can be used to compare and show the differences between amino acid or nucleotide sequences of different enzymes or the genes that code for them. Besides, we can predict, visualize, and model the structure of the enzyme. Some software can show you how a polypeptide folds into its 3D shape using its amino acid sequence or the DNA sequence of its gene. Lastly, scientists can search the database to identify restriction endonucleases to use in their research. They can also discover new enzymes which might be suitable for their experiments in the database. D. Compare the similarities and differences between electrophoresis and paper chromatography. Both techniques are separating techniques which involve the movement of substances in a mixture. However, electrophoresis is used to separate DNA, RNA, and protein. Chromatography is used to separate pigments in a plant extract. The stationary phase is not the same. They are agarose gel and paper. All separation methods have a stationary and mobile phase. The stationary phase is the one that does not move with the sample. The separation in electrophoresis is done by electric current, while chromatography is done by solvents such as acetone. In electrophoresis, DNA fragments are separated due to their charges and lengths. Smaller and lighter fragments travel further in the gel. In chromatography, pigments are separated due to their solubilities and mass. The greater the solubility in the solvent, the further the pigment can travel on the paper. To visualize DNA, we can use dye or ethidium bromide. Ethidium bromide is a substance that can bind with DNA. When exposed to UV light, it will fluoresce with an orange color. In chromatography, pigments can normally be visualized by their colors. To identify a specific DNA, we can use a DNA probe. A probe is a single-stranded DNA or RNA used to search for its complementary sequence in a sample genome. In chromatography, pigments are identified using RF values. RF value is calculated by the distance traveled by the pigment divided by the solvent front. 5a is about warfarin resistance. 1 wants you to use the Hardy-Weinberg equations to calculate the percentage of the rats that is heterozygous for warfarin resistance. P and Q in equation 1 represent the allele frequency of the dominant and recessive alleles for this trait. P squared in equation 2 is the frequency of the homozygous dominant individuals. 2pq is the heterozygous, while q squared is the homozygous recessive individuals. The question says that warfarin resistance is due to a dominant allele. It also gives the percentage of rates that have the resistance. Those are the homozygous dominant and heterozygous individuals. The remaining 60% of rats do not have resistance. They are the homozygous recessive individuals. This means q squared is 0.6. Q is the square root of 0.6. The recessive allele frequency is 0.775. By using equation 1, we now know that P is 0.225. The frequency of heterozygous individuals is 2pq. The answer is 0.35, which means it is 35%. 2. Suggest and explain which of the conditions of the Hardy-Weinberg principle are most likely to be met for the island population of brown rates. The Hardy-Weinberg principle states that a population's genetic variation will remain constant from one generation to the next without any disturbing factors. It assumes that there is no migration, which can cause the flow of alleles into and out of the population. Since the rats are found on an island, they are isolated from the other populations, so this condition is likely to be met. Random mating must occur. If the individuals with certain characteristics have a higher chance of mating, the allele codes for this trait will keep increasing. Equilibrium cannot be maintained. Besides, the rates move around freely on the island. This causes all members of the population to have a chance to mate randomly. The rat population is large. Genetic drift likely has little effect on the gene pool and hence will not alter the allele frequencies from time to time. The rates are diploid. For polyploid organisms which have more than 2 sets of chromosomes, the equations have to be expanded further. The ones that are shown in these questions are only for organisms that are either homozygous dominant, heterozygous, or homozygous recessive. Lastly, since there are only P and Q in the equation, this is only for a gene with 2 alleles. B wants you to explain why when a new dominant advantageous allele occurs, its frequency increases more quickly in the population than a new recessive advantageous allele. A new allele appears when there is a mutation. When a mutation occurs in an organism where one copy of the gene is altered, it becomes a heterozygous individual. So, a new allele will initially be found in heterozygotes. The new dominant allele is always expressed. The organism will show the beneficial phenotype coded by the mutated gene. Selection or environmental conditions can act straight away on the individuals with the dominant allele, increasing its frequency. On the other hand, if the new allele is recessive, it is not expressed in heterozygotes. You can only get an individual with the characteristics when a homozygous recessive genotype occurs. So, selection would not start right after the mutation. It can only act when homozygous recessive individuals begin to appear. These will take time, leading to a slower increase in the allele frequency. In 6a, we have a photomicrograph of a section through a Bowman's capsule and a glomerulus. The glomerular filtrate is found in the capsular space. It is the clear space inside the Bowman's capsule. Blood plasma is inside the capillaries, which forms the glomerulus. Remember to use a label line for your answer. You won't get a mark if you write the letter on the diagram without a label line. B shows the mean GFR for healthy males and females of different age groups. 1. Calculate the yearly rate of decrease in mean GFR from the 20 to 29 age group to the 80 to 89 age group for females. GFR decreases from 118 to 52. Find the difference between them and divide them by the total age gap, 69. If you use the lowest or highest age in the groups, it is acceptable as well. 2. Suggest and explain other factors that can affect GFR. It can be affected by the hydrostatic or blood pressure. If the pressure is higher, ultrafiltration occurs more rapidly and GFR will be higher. Water and solute potential gradient between glomerulus and Bowman's capsule can affect GFR, and it will determine the rate of osmosis. Some kidney diseases can reduce the efficiency of nephrons and decrease GFR. Damages to the nephron or basement membrane can cause more filtrate to be formed in a shorter time, as water and other substances can pass through more easily. Dehydration can lead to a lower water potential of blood. With less water in the blood, glomerular filtrate forms more slowly. High altitude decreases GFR. This is because our bodies compensate for the low partial pressure of oxygen by increasing the number of red blood cells. Renal blood flow will decrease. Some drugs, such as diuretics and vasodilators, may affect GFR. A larger difference between afferent arterial lumen and efferent causes higher GFR. The greater the difference, the higher the hydrostatic pressure is in the glomerulus. C. Describe the process of selective reabsorption. First, sodium ion is actively transported out of the PCT cells. ATP is required for this movement. It will then enter the blood. This movement causes its concentration in PCT cells to decrease. A concentration gradient between the PCT lumen and the cytoplasm of PCT cell is created. Sodium ion enters cell from filtrate or lumen by facilitated diffusion down its concentration gradient. It moves through the co-transporter proteins. This leads to the co-transportation of glucose and amino acids. Glucose and amino acid concentrations increase in the cell. They will leave the cells by facilitated diffusion. Glucose and amino acids will then be reabsorbed into the tissue fluid and then into the blood. The movement of these solutes into the cells causes the water potential of PCT cells to decrease. Water enters PCT cells from the filtrate by osmosis down the water potential gradient. There is a mark for AVP. For example, you can name other ions that are reabsorbed. You can also name the transport protein for the movement of glucose out of the PCT cell. It is called a GLUT transporter. 7a says that the efficiency of striated muscle contraction decreases when the air temperature decreases. You are asked to suggest the reasons. Respiration is an enzyme control reaction. A lower temperature reduces the activity of enzymes in respiration as there will be less kinetic energy. This causes less ATP to be produced. At a lower temperature, calcium ions, sodium ions, and ACH movement is reduced. This is because they have a lower kinetic energy when the temperature is low. Sodium ion is required for action potential. Calcium ion is needed to cause the exocytosis of acetylcholine. Due to a lower enzyme activity, less acetylcholine is broken down in the neuromuscular junction. The enzyme that catalyzes this reaction, acetylcholinesterase, is less active due to its low kinetic energy. If acetylcholine is not broken down, it remains attached to the receptors. In the sarcomere, there will be fewer calcium ions moving around and bind to troponin. Again, it is due to its lower kinetic energy. This leads to fewer cross bridges being formed, and fewer power strokes as the binding is required to expose the myosin binding site on actin. ATP is required for the detachment of myosin heads from actin. When the rate of ATP synthesis is lower, there will be fewer cross bridges being broken. This is also due to the lower activity of ATPase, the enzyme on myosin head that hydrolyzes ATP. Without the detachment, we can't have the next stimulation of muscle contraction. You can get a mark for AVP if you talk about the other aspects such as blood flow and thermoregulation. B talks about a neurotoxin called TTX. It binds to voltage-gated sodium ion channels in the axon of a neuron and changes the tertiary structure of the channel protein. Suggests how TTX may affect the functioning of a motor neuron. There are two possible ways. First, the sodium ion channels don't open due to the binding. Sodium ion does not diffuse into the neuron. Without this diffusion, the inside of the membrane cannot become more positive. Axon membrane cannot be depolarized. there will be no action potential being generated. In other words, the impulse is not transmitted along the motor neuron. Without an action potential, calcium ion channels at the synapse would not open. Calcium ion does not enter the presynaptic node. They are needed to stimulate the acetylcholine-containing vesicles to move and fuse with the presynaptic membrane, leading to the release of acetylcholine by exocytosis. This can cause paralysis, the loss of voluntary muscle function. Another possibility is that the shape change causes sodium ion channels to stay open. Sodium ion always diffuses into the neuron. The inside of the membrane is always positive, so the membrane stays depolarized. A continuous action potential is generated. At the synapse, calcium ions always enters the presynaptic node. Vesicles are always stimulated to move and fuse with the membrane. Acetylcholine is continually released by exocytosis. This causes continued muscle contraction. In A, we have a graph that shows the rate of activity of Rubisco in non-GM rice and GM rice at different concentrations of atmospheric carbon dioxide. A1, compare the two curves and explain why the curve for non-GM rice levels off. The command words compare means that you should talk about both the similarities and the differences. At low carbon dioxide concentrations, the rate of activity for both increases. However, the rate is higher in GM rice at all the carbon dioxide concentrations. Quoting in comparative figures to support the second point can give you a mark. For example, you can quote the rate of activity of both when carbon dioxide concentration is 200 mg per dm cube. Remember that you must write the correct unit for the values. The graph levels off at high carbon dioxide concentrations because it's no longer limiting. You can also get this mark by detailing why it is no longer a limiting factor. For example, you can explain that the active sites of Rubisco are saturated with carbon dioxide. You can also talk about the other factors that are now limiting the reaction. To increase the rate of activity, you have to increase these factors. Another reason is that there is less RUBP available as most of them have already reacted with carbon dioxide. 2. It is the enzyme's active site that is altered, which causes a change in its ability to catalyze the reaction. B1. Name the process that involves the enzyme Rubisco. This is the enzyme that catalyzes carbon fixation, where carbon dioxide is fixed to RUBP, forming an unstable 6-carbon compound. GP receives hydrogen from reduced NADP to become TP. So, this is a reduction by hydrogenation. For reduced NADP, it is oxidation by dehydrogenation. 10 molecules of TP have 30 carbons in total. RUBP has 5 carbons, so we can make 6 molecules of it. TP can be used to make all kinds of photosynthetic products. Carbohydrates, including monosaccharides, disaccharides, and polysaccharides, can be synthesized. My suggestion is to use specific names of carbohydrates such as glucose, sucrose, and starch instead of the common term to answer this question. Amino acids and the components of lipids can also be synthesized. The endocrine system consists of all the endocrine glands in our bodies. Glucagon is a protein hormone. All protein hormones are hydrophilic. They cannot pass through the hydrophobic region of the phospholipid bilayer. So, their receptors are located on the cell surface membrane. The binding of the hormone with its specific receptor triggers a second messenger pathway. Second messenger, such as CAMP, will be produced in the cell. Insulin has 3 target cells: liver cells, muscle, and adipose cells. ADH has 2, the DCT cells and the collecting duct cells. These are the cells involved in the reabsorption of water from the filtrate back to the blood. The maintenance of a constant internal environment is known as homeostasis. In B, we have a graph that shows how blood glucose levels change after a glucose meal. B1 wants you to explain how the blood glucose concentration is reduced during phase A. Following a glucose meal, blood glucose concentration increases above the set point. This is considered as a stimulus. The stimulus leads to the production and release of insulin by the beta cells in the pancreas. Insulin is released into the blood and travels to the target cells. It will stimulate the cells to increase the uptake of glucose into them. In muscle and adipose cells, a transport protein called GLUT will be added to their cell surface membrane. An increased number of GLUT on the cell surface membrane leads to an increase in the permeability to glucose. In muscle cells, glucose will be phosphorylated. There will be an increase in the rates of respiration using glucose as the substrate. In adipose cells, glucose molecules that are taken up are being converted into lipids. In liver cells, phosphorylation of glucose occurs. The rate of glycogenesis increases where glucose is being used to synthesize glycogen. 2. Suggests why the blood glucose concentration increases again during phase B. Phase B is about 2.5 hours after the glucose meal, so the increase is not because of glucose consumption. The insulin action causes the blood glucose concentration to drop below the set point. This will in turn stimulate the release of glucagon by alpha cells. Glucagon leads to an increase in blood glucose concentration. A. Outline the role of zoos in the conservation of endangered species other than captive breeding programs. The zoo provides a safe location for the animals where they are protected from predators, hunters, and poachers. Zoos also undergo research on disease control, diet, and behavior. Conservation can be more effective by having a better understanding of the animals. For this point, you have to elaborate on the types of research being done. Carrying out research alone will not give you any marks. They can help to raise funds for the conservative efforts, as many of these programs are very costly. If you only talk about raising funds without any elaboration, you may not get this mark. Zoos can work with local communities and governments to organize conservation projects. They can educate and raise the awareness of the public on the factors they become endangered. This can help the visitors to understand their roles and responsibilities in the conservation effort. Lastly, zoos can provide veterinary care to the animals to increase their lifespan and quality of life. B wants you to suggest reasons why fewer female black rhinos breed in captivity than in the wild. Captivity is a simulation of the natural habitat, but it cannot be completely identical. So, it is an unnatural environment for the animals. This can bring about hormonal and physiological changes that have negative impacts on fertility. Some animals might have behavioral changes and refuse to mate. If they do not show courtship behavior, breeding will not be successful. The number of animals in captivity is very limited. So, the availability of suitable mating partners may be low. Some animals experience stress and pressure being kept in captivity. Stress might cause us unhealthy weight loss and gain, a suppressed immune system, reproductive failure, and general physiological distress. Lastly, the program is limited by the zoo's capacity. They often need to manage the breeding numbers to avoid exceeding the maximum capacity. C outlines the process of IVF that is used in captive breeding programs. After oocytes are collected, they are incubated with semen for fertilization to take place. The embryos produced are not necessarily implanted back into the mother. A surrogate mother can be used sometimes. Surrogate is a female who becomes pregnant for the purpose of carrying the fetus to term for the biological mother. Cryopreservation can also be done for future implantation. It is a process that preserves organelles, cells, tissues, or any other biological constructs by cooling the samples to very low temperatures. That's all for today. If you think my videos are useful, don't forget to like, comment, and subscribe. Thank you for watching and see you again soon.